Greenwich, London, site of 0 degrees longitude. Geneva, Switzerland, home city of the CERN Large Hadron Collider. Just as the Greenwich Observatory is considered the home of time, and CERN the heart of nuclear physics, so this campus on the edge of Cambridge is forever linked with the science of genomics. The Wellcome Genome Campus is where the first animal genome of a simple nematode worm was sequenced in the late 1990s. And now the campus is leading a revolution in genomics and biodata, bringing the prospect of previously unimagined medical breakthroughs. So, scientists from all over the world are attracted to work here. Let's see how they define what a genome is.
I would say the genome is a blueprint of all the information needed to generate a whole organism.
Every organism has its own genome. Yeah. Just as the genome connects us with all other humans. You know, we all share much more of our code than we have that is different.
Not only between each species is the genome different, but actually each individual of that species has a slightly different genome.
It's those instructions that define how the natural world looks.
That is much better than I can explain.
So, every living thing has a genome, a code carried in each cell, spelled out in DNA. Not necessarily an easy concept, which is perhaps why most people around the world barely blinked when this campus was established in the early 90s. 10 years later, it was a different story, when the Human Genome Project, a global collaboration based here and in America, announced, ahead of time and under budget, mission accomplished.
Scientists on both sides of the Atlantic said today they'd completed a rough draft of the entire human genetic code.
That would have seemed like science fiction 15 years ago.
A revolution in medical science whose implications far surpass even the discovery of antibiotics.
In coming years, doctors increasingly will be able to cure diseases like Alzheimer's, Parkinson's, diabetes, and cancer.
The Prime Minister and President Clinton have joined forces to call on scientists to give free access to their discoveries on the makeup of the human genetic code.
The foundation of this institute is to enable that data to be made globally available for human health.
Pushing an open access agenda ensures that everyone gets to work with that data irrelevant of the amount of money they have.
And what's the point of knowing something if it's hidden in a cupboard somewhere and no one else can access it?
Sequencing DNA samples from humans, animals, or indeed from bacteria, viruses, or parasites now takes place here on a staggering scale, assisted by robots and computers. The quantities of data generated are breathtaking. The human genome is made up of 3.2 billion letters. If printed out, these would stretch from here to the Sahara Desert. From person to person, our genomes differ by just 0.5%, a tiny fraction, which still amounts to millions of differences. These vast datasets have fueled a whole new branch of computer-driven biology called bioinformatics.
Biology used to be thought of as quite a soft science. You know, there were lots of pictures, there were lots of images, there were lots of descriptions. And now it's moving in a completely different direction. It's about data and analytics.
We really need the computers to be able to understand the complexity of the data.
And it's not just kind of a standard computer these days either. You couldn't just do it on your kind of desktop computer or your PC. You really need very special infrastructure.
When you have an entire floor that's dedicated to just petabytes and petabytes of servers and storage, you know what you're dealing with.
So we're talking about storage for an ever-increasing archive of genomes, with the speed of growth relentless. Just think, it took 13 years to sequence the first human genome. Now, machines in our sequencing labs produce the equivalent of a human genome every half hour, enabling biology on a scale like nowhere else. But all this knowledge only becomes really interesting when it's put to work, comparing genomes to spot similarities and differences. Nowhere is this more important than in the field of infectious disease, where comparing the genomes of different bacteria is already yielding life-saving results.
We can identify MRSA, which is a bacteria that's quite a problem in some hospitals. We can identify transmission of MRSA from person to person.
All salmonella genomes are now routinely sequenced at Public Health England, and they've moved away from traditional methods, which are laboratory-based for identifying particular lineages or strains, to move fully towards genome sequencing everything.
All genomes evolve over time, each generation changing slightly. For humans, with 25 years between generations, the rate of change is slow. But for bacteria or viruses, whose generation time may be just hours, evolution is rapid. That's how antibiotic resistance can emerge. So tracking genome changes is key to developing effective drugs.
The ability to predict how pathogens will evolve and how they will mutate and change is something that's really powerful, especially when it comes to, say, viruses, which are constantly changing.
One infectious disease still kills nearly 500,000 people every year, mostly children in Africa. Malaria is caused by a parasite carried by mosquitoes. There are high hopes that knowledge gleaned from a combined approach of sequencing parasites, mosquitoes, and humans will lead to breakthroughs.
I believe that hopefully will spell the end of malaria, and that really eradication is our goal.
Do you think that's a realistic prospect within 5-10 years?
I think we're looking beyond 10 years, unfortunately. But yes, I'm hoping in my lifetime.
And within our lifetimes, what else should we expect?
You'll go to the doctor and they'll just say, right, this is what's wrong with you. We can match what's wrong with you with these antibiotics or this particular type of medicine, and away you go. Do you not think that's...
I think that's very optimistic. I would love us to get there, but I think human genome is so complex that it will take us a long time to really understand how each individual variation in our genome translate into some effect.
And large-scale DNA sequencing is also shedding light on a disease that affects 1 in 3 of us. Routine sequencing of tumours is completely changing how we'll diagnose and treat cancer.
In a cancer genome, we see about 10,000 mutations, but maybe only 1 to 10 of those are causal for the cancer.
But by identifying these key mutations, you can start to tailor clinical interventions that have the best chance of outcome for these patients.
So we didn't know that there are different types of breast cancer and that they have to be treated in a different way. And now we know that, and we can also use genomics to inform us about how to treat the cancer.
Genomics is changing our definition of cancer, just in the same way that genomics is changing our definition of many different diseases.
Scientists here are using the most advanced technologies available to deliver results on an unprecedented scale. From parasites to people, the secrets locked in individual genomes are being revealed.
We're moving closer and closer to discovering more and more about ourselves, some of which we want to discover and some of which we don't, I'm guessing.
That's interesting. I think, so to me, it's mind-blowing how fast we're going, how fast we're advancing in this field.
This is not just about diseases that affect rich people or the developed world.
Sequencing technology is increasingly getting cheaper and more portable.
It's really important that it's explained exactly what the data is going to be used for and how it will be treated.
And for example, I wouldn't want my insurance to know how prone I am to heart disease or something similar.
Personally, I would have no issue at all publishing my genome and letting people tell me things about myself I didn't already know.
Would you want to know the genetic information of a baby? Should they be sequenced at birth?
Definitely, I think I would like to have my baby's genome sequence?
I think I might wait until they're old enough to make the decision themselves.
Gene editing is really exciting and the potential there is just mind-boggling, but do you not think we have to be a bit careful and just make sure we fully understand absolutely everything before this is implemented?
If you don't talk to the scientists, you're not really going to get a feel for what actually goes on here and how we do our work. So I think that's the best way, really, to understand at the campus and what we do.
Everyone has to discuss it together.
And so I think it's our fundamental responsibility to help people understand what genomics means, what the potential is, but also more than just explain, to listen.
Even though we are still quite a long way off from doing some of these things that would raise very serious ethical concerns, I think you have to have that debate before we get there.
We hope you'll share our excitement about what lies ahead and be involved in discussions about where this research should go.